Photosynthesis. Photosynthesis is the process by which green plants, algae, and some bacteria convert light energy from the sun into chemical energy in the form of glucose, sugar. During photosynthesis, the chlorophyll pigment in the chloroplasts of the plant cells captures light energy and uses it to split water molecules into oxygen and hydrogen ions. The oxygen is released into the atmosphere, while the hydrogen ions combine with carbon dioxide CO2 from the air to produce glucose, C6H12O6. This process occurs in two main stages, the light-dependent reactions and the light-independent reactions, also called the Calvin cycle. In the light-dependent reactions, light energy is used to create energy-rich molecules, such as ATP and NADPH, which are used in the light-independent reactions. In the light-independent reactions, the energy-rich molecules are used to convert CO2 into glucose. Photosynthesis is vital for the existence of life on Earth, as it provides the primary source of food and oxygen for living organisms. External parts of a leaf and their functions. The external parts of a leaf and their functions are as follows. 1. Blade, lamina. The blade is the flattened, expanded part of the leaf that is responsible for most of the photosynthesis. 2. Petiole. The petiole is the stalk that attaches the blade to the stem and allows the blade to move to optimize light exposure. 3. Veins. The veins are the channels that provide a framework for the transport of water, minerals, and sugars to and from the blade. 4. Stipules. The stipules are a pair of small, leaf-like appendages at the base of the petiole that help protect the newly forming leaves. 5. Cuticle. The cuticle is the waxy layer on the upper epidermis of the leaf that helps reduce water loss by evaporation. 6. Epidermis. The epidermis is the outermost layer of cells that provides protection for the leaf. 7. Guard cells. The guard cells are two specialized cells that form a pore, called a stomata, which helps regulate the exchange of gases and water vapor between the leaf and the atmosphere. 8. Spongy mesophyll. The spongy mesophyll is the layer of loosely packed cells that lies just below the palisade mesophyll and allows for gaseous exchange. 9. Palisade mesophyll. The palisade mesophyll is the layer of elongated, tightly packed cells that lies just under the upper epidermis and is responsible for most of the photosynthesis. Together, these parts of a leaf allow it to optimize photosynthesis by controlling the flow of water, minerals, and nutrients, and regulating the exchange of gases and water vapor. Internal parts of a leaf and their functions. 1. Epidermis. The epidermis is the outermost layer of cells in a leaf, and it serves several functions. 1. Protection. The epidermis helps protect the leaf from damage, disease, and pests. 2. Gas exchange. The epidermis contains stomata, which are small openings that allow for the exchange of gases such as carbon dioxide and oxygen with the atmosphere. 3. Regulating water loss. The presence of stomata in the epidermis also allows for some water vapor to escape as part of transpiration, but the presence of the cuticle, a waxy layer on top of the epidermis, reduces the amount of water lost through the leaf surface. 4. Secretion. Some epidermis cells can secrete substances such as trichomes, tiny hairs or outgrowths that can help reduce water loss, protect the leaf from herbivores, and reflect sunlight in areas of high solar radiation, or nectar that attracts pollinators. Overall, the epidermis supports and protects the internal tissues involved in photosynthesis, water movement, and nutrient transport within the leaf. 2. Guard cells. Guard cells are specialized cells found in the epidermis of a leaf that surround and control the opening and closing of stomata. They play a crucial role in regulating gas exchange and water balance in the leaf. When the guard cells are turgid swollen with water, they cause the stomata to open, allowing for the exchange of gases such as carbon dioxide, oxygen and water vapor. This process promotes photosynthesis and transpiration, which is essential for plant growth and survival. When the guard cells are flaccid deflated, they cause the stomata to close, reducing water loss during dry conditions and preventing the entry of pathogens. The opening and closing of the stomata is regulated by the movement of ions such as potassium, which is pumped into the guard cells causing water to follow by osmosis. This change in water volume causes the guard cells to either swell or deflate, opening or closing the stomata respectively. Overall, guard cells play an important role in maintaining water balance and gas exchange in the leaf, responding to changing environmental conditions such as light, temperature, humidity, and water availability. 3. Palisade layer. The palisade layer, also called palisade mesophyll, is a layer of elongated, closely packed cells found in the upper part of the mesophyll inside a leaf. It is responsible for a crucial process in photosynthesis and provides support to the plant tissue. The functions of palisade layer include 1. Photosynthesis. The palisade layer contains the majority of the chloroplasts which convert light energy from the sun into chemical energy in the form of sugar. These chloroplasts contain pigments such as chlorophyll that absorb light, and the high density of chloroplasts in the palisade layer allows for efficient capture of light energy. 2. Gas exchange. The palisade layer allows gaseous exchange to occur as it is located close to the upper surface of the leaf where stomata are found. Carbon dioxide CO2 enters through the stomata located in the lower epidermis and diffuses through the mesophyll to reach the palisade layer. 3. Structural support. The tightly packed cells of the palisade layer provide structural support to the leaf, which helps to keep it upright and efficient in capturing sunlight for photosynthesis. Overall, the palisade layer plays a vital role in photosynthesis by facilitating the conversion of light energy into chemical energy, and is also involved in regulating gas exchange and providing structural support to the leaf. 4. Spongy mesophyll layer. The spongy mesophyll layer is a layer of loosely arranged or irregularly shaped cells in the lower part of the mesophyll inside a leaf. It has several important functions. 1. Gas exchange. The spaces between the spongy mesophyll cells allow for the diffusion of gases such as oxygen and carbon dioxide between the air spaces in the leaf and the rest of the internal tissues of the leaf. 2. Photosynthesis. Although there are fewer chloroplasts in the spongy mesophyll layer compared with the palisade layer, they are still present and can carry out photosynthesis, contributing to the overall photosynthetic activity of the leaf. 3. Storage. The spongy mesophyll layer can also function as a storage site for excess sugars and other nutrients produced during photosynthesis. 4. Support. The spongy mesophyll layer helps to provide support and resilience to the leaf, especially during changes in air pressure or water availability. Overall, the spongy mesophyll layer plays an important role in gas exchange, photosynthesis, and storage, while also providing support to the leaf tissue. Its loose, sponge-like structure helps to maximize the exchange of gases and nutrients within the leaf. 5. Leaf veins. Leaf veins are the structures that support and transport fluids, water and nutrients, throughout the leaf. They consist of two types of vascular tissue, xylem and phloem. 1. Xylem. The xylem tissue in leaf veins transports water and dissolved minerals from the roots of the plant to the rest of the leaf. It is made up of large, hollow, tube-like cells called vessels, as well as smaller, elongated cells called tracheids. 2. Phloem. The phloem tissue in leaf veins transports sugars and other organic compounds produced during photosynthesis to the rest of the plant. It is made up of specialized cells called sieve tubes, along with companion cells that provide metabolic support. The veins of a leaf also provide structural support to the leaf and help to distribute water and nutrients evenly throughout the leaf tissue. Additionally, the arrangement of veins in a leaf can affect its overall shape and structure, with different types of veining patterns contributing to the diversity of leaf shapes seen in plants. 6. Chloroplast. Chloroplasts are specialized structures found in the cells of green plant leaves and other photosynthetic organs. They are responsible for carrying out the process of photosynthesis, converting light energy into chemical energy that can be used by the plant for growth and metabolism. Chloroplasts have several important parts, including 1. Thylakoid membrane, a system of interconnected membranes that contain the chlorophyll pigments and other molecules required for photosynthesis. The thylakoid membrane is arranged in stacks called grana, which help to increase the surface area available for light absorption. 2. Stroma, a fluid-filled space surrounding the thylakoid membranes, where the reactions that convert light energy into chemical energy take place. 3. Chlorophyll pigments, molecules that absorb light energy and use it to drive photosynthesis. Chlorophyll gives plants their green color. 4. Electron transport chain, a series of electron-carrying molecules that pass electrons from chlorophyll to other molecules, generating a proton gradient that powers the production of ATP. Overall, chloroplasts are essential organelles for photosynthesis in plants, allowing them to convert sunlight into the energy they need to survive and grow. Requirements and conditions necessary for photosynthesis. Photosynthesis is the process by which green plants, algae, and some bacteria convert light energy into chemical energy in the form of organic compounds, such as glucose. The process requires certain environmental conditions and specific cellular structures in order to occur efficiently. 1. Light energy. Photosynthesis requires a source of light, typically sunlight. The light energy is absorbed by chlorophyll pigments in the chloroplasts of plant cells. 2. Carbon dioxide CO2. Carbon dioxide is taken in from the atmosphere through small openings on the leaves called stomata. It is used as a carbon source for the production of organic compounds. 3. Water H2O. Water molecules are taken up by plant roots and transported to the leaves through the xylem tissue. They are split by photosystem II during the light-dependent reactions, releasing oxygen gas as a byproduct. 4. Chloroplasts. Chloroplasts are organelles found in the cells of green plants and algae that carry out photosynthesis. They contain pigments such as chlorophyll, that capture light energy, as well as enzymes and other molecules required for the chemical reactions of photosynthesis. 5. Enzymes and other molecules. Photosynthesis requires a variety of chemical reactions to occur, including the conversion of CO2 and water into glucose and other organic compounds. These reactions are catalyzed by specific enzymes and require a range of other molecules such as ATP, NADPH, and carbon fixation enzymes. In addition to these basic requirements, certain conditions can either enhance or limit photosynthesis, including factors like temperature, light intensity, and availability of nutrients, such as nitrogen and phosphorus. These variables can affect the rate of photosynthesis and the overall productivity of plant growth. The process of photosynthesis. Photosynthesis is the process by which green plants, algae, and some bacteria convert light energy into chemical energy in the form of organic compounds, such as glucose. It occurs in two main stages. 1. Light-dependent reactions. The first stage of photosynthesis occurs in the thylakoid membranes of the chloroplasts. Light energy is absorbed by chlorophyll pigments and used to generate ATP and NADPH, which are energy-rich molecules that are used in the second stage. Water molecules are also split, releasing oxygen gas as a byproduct. 2. Light-independent reactions. Calvin cycle. The second stage of photosynthesis occurs in the stroma of the chloroplasts. Carbon dioxide molecules enter the cycle, which uses energy from ATP and NADPH to drive the synthesis of organic compounds, such as glucose. The Calvin cycle also involves a series of chemical reactions that recycle carbon dioxide to keep the cycle going. Overall, photosynthesis converts light energy into chemical energy in the form of organic compounds, while also producing oxygen gas as a byproduct. The process is essential for the survival and growth of green plants and other photosynthetic organisms, as it provides them with the energy they need to carry out cellular processes and build new tissues. Equation of photosynthesis. The equation for photosynthesis is 6CO2 plus 6H2O plus light energy, C6H12O6 plus 6O2. This equation shows the overall process of photosynthesis, in which carbon dioxide CO2 and water H2O are converted into glucose, C6H12O6, and oxygen gas O2 in the presence of light energy. The process involves two main stages: the light-dependent reactions, which take place in the thylakoid membranes of chloroplasts, and the light-independent reactions, Calvin cycle, which take place in the stroma of chloroplasts. During the light-dependent reactions, light energy is absorbed by chlorophyll pigments and used to generate ATP and NADPH, which are energy-rich molecules that are used in the light-independent reactions. Water molecules are also split, releasing oxygen gas as a byproduct. During the light-independent reactions, carbon dioxide molecules enter the Calvin cycle, which uses energy from ATP and NADPH to drive the synthesis of organic compounds, such as glucose. Overall, the equation for photosynthesis represents the conversion of light energy into chemical energy in the form of organic compounds, while also producing oxygen gas as a byproduct. End products of photosynthesis. The end products of photosynthesis are glucose, C6H12O6, and oxygen gas O2. Glucose is a simple sugar that is used by plants and other photosynthetic organisms as a source of energy to carry out cellular processes and to build new tissues. Oxygen gas is released as a byproduct of photosynthesis and is essential for respiration in living organisms, including plants, animals, and humans. During respiration, oxygen is used to break down glucose and other organic compounds, releasing energy in the form of ATP, which is used to power cellular processes. Therefore, photosynthesis and respiration are essential processes that are closely connected and interdependent in maintaining the balance of life on Earth. Practical activity. To test for starch in a leaf. Show the procedures and results of experiment. Procedure. 1. Take a green leaf from a plant and place it on a clean surface. 2. Add a few drops of iodine solution onto the leaf. 3. Wait for about 1 to 2 minutes to observe the change in color. Results. If the leaf contains starch, the part of the leaf covered with iodine will turn blue-black in color. This is because iodine reacts with starch to form a blue-black complex. If the leaf does not contain starch, the color of the leaf will not change, and it will not turn blue-black. Explanation. Starch is a complex carbohydrate that is produced by plants during photosynthesis. It is stored in the leaves, roots, and other parts of the plant as a source of energy. Iodine is used as a test for starch because it reacts with the glucose units in starch to form a blue-black complex. When iodine is added to a leaf that contains starch, it will react with the starch in the leaf to form a blue-black complex, indicating the presence of starch. If the leaf does not contain starch, iodine will not react, and the color of the leaf will not change. Note: the experiment can also be carried out by placing the leaf in boiling water for a few minutes to soften the tissues and remove any chlorophyll present. The leaf is then rinsed in cold water to cool it down before adding iodine solution to test for the presence of starch. Practical activity. Aim: to investigate the gas released during photosynthesis. Give the procedures and results of the experiment. Procedure 1. Fill a beaker with water and place an aquatic plant, such as Elodia, in the beaker. Ensure that the plant is fully submerged in the water. 2. Place the beaker under a light source, such as a lamp or sunlight. 3. Leave the plant in the light for about 10 to 15 minutes to allow photosynthesis to occur. 4. Collect the gas bubbles that are released by the plant by inverting a small test tube filled with water over the plant. 5. Leave the test tube for about 10 to 15 minutes to allow the gas bubbles to collect at the top of the tube. 6. Test the collected gas for the presence of oxygen using a glowing splint test. Results: if photosynthesis occurs, the gas bubbles collected should be mostly oxygen gas O2. This can be tested by using a glowing splint to check for the presence of oxygen. When a glowing splint is inserted into the test tube, the splint will reignite if oxygen is present, indicating the release of oxygen by the plant during photosynthesis. Explanation: during photosynthesis, plants absorb carbon dioxide CO2 from the atmosphere and water H2O from their surroundings, and release oxygen gas O2 as a byproduct. The process requires light energy, which is absorbed by pigments such as chlorophyll in the plant's leaves. The light energy is used to convert the absorbed carbon dioxide and water into glucose, C6H12O6, a simple sugar that is used by the plant as a source of energy, and oxygen gas O2, which is released into the atmosphere. The experiment described above allows us to collect the oxygen gas released by the plant during photosynthesis and test for its presence using the glowing splint test. Note: the experiment can be modified by using a gas syringe to collect the gas bubbles instead of a test tube. The collected gas can also be tested for the presence of carbon dioxide using lime water, KO2, to confirm that the gas bubbles collected are mostly oxygen. Adaptation of the leaf to the process of photosynthesis. Leaves are the main organs of photosynthesis in plants, and they are adapted to maximize the efficiency of this process in the following ways. 1. Large surface area. Leaves have a large surface area compared to their volume, which allows them to absorb as much light as possible for photosynthesis. 2. Thin shape. Leaves are thin and flattened, which reduces the distance that light must travel to reach chloroplasts in the cells where photosynthesis occurs. 3. Broad and flat shape. The broad and flat shape of leaves helps to expose chloroplasts to sunlight at different angles during the day, thereby increasing the rate of photosynthesis. 4. Chloroplasts. Chloroplasts are specialized organelles found in plant cells that contain chlorophyll pigments. These pigments absorb light energy for photosynthesis, and the presence of many chloroplasts in leaf cells increases the amount of photosynthesis that can occur. 5. Stomata. Leaves have small pores called stomata that allow for the exchange of gases between the leaf and the surrounding air. Carbon dioxide can enter the leaf through the stoma, and oxygen is released from the leaf through the same pores. 6. Veins. Leaves have a network of microscopic veins that transport water and nutrients to cells throughout the leaf. This helps to maintain the high rates of photosynthesis by providing the necessary raw materials for the process. These adaptations help to maximize the rate of photosynthesis in leaves and ensure that plants can produce enough energy to survive and grow. Factors affecting the rate of photosynthesis. The rate of photosynthesis in plants can be affected by several factors, including 1. Light intensity. As the intensity of light increases, the rate of photosynthesis also increases, up to a certain point where the plant becomes saturated and further light does not increase the rate of photosynthesis. 2. Carbon dioxide concentration. As the concentration of carbon dioxide in the air around the plant increases, the rate of photosynthesis also increases, up to a certain point where the plant becomes saturated and further carbon dioxide does not increase the rate of photosynthesis. 3. Temperature. Photosynthesis is a chemical process that is sensitive to temperature. As the temperature increases, the rate of photosynthesis increases up to a certain point where it becomes too hot for the enzymes to function properly, leading to a decrease in the rate of photosynthesis. 4. Water availability. Photosynthesis requires water, and a lack of water can lead to stomata closing and the rate of photosynthesis decreasing. 5. Chlorophyll concentration. If the plant has insufficient chlorophyll due to a nutrient deficiency, disease or damage to the plant, then the rate of photosynthesis may be affected. These factors can interact with each other to affect the rate of photosynthesis. For example, on a hot day, a plant may close its stomata to conserve water, which would decrease the rate of photosynthesis. Understanding how these factors affect photosynthesis is important in agriculture and horticulture for optimizing crop growth and productivity. Importance of photosynthesis. Photosynthesis is of critical importance to life on Earth. Here are some reasons why. 1. Oxygen production. Photosynthesis is responsible for producing most of the oxygen in Earth's atmosphere, which is essential for the survival of animals and humans. 2. Food production. Photosynthesis is the primary source of energy for most life on Earth. Plants use it to produce glucose, which is then consumed by other organisms in the food chain. 3. Carbon fixation. Photosynthesis involves the fixation of carbon dioxide into organic molecules like sugars, which is essential for maintaining the balance of carbon in the atmosphere and for mitigating climate change. 4. Energy production. Photosynthesis is the basis of most renewable energy sources such as solar, wind, and hydropower. 5. Ecosystem services. Photosynthesis supports a wide range of ecosystem services such as carbon sequestration, nutrient cycling, water purification, and soil conservation. In essence, photosynthesis is the foundation of life on Earth, supporting both plant and animal life as well as the physical environment. Practical activity. Aim, to test the importance of chlorophyll during photosynthesis. Give the requirements, procedures and results. Requirements. A healthy green leaf. A healthy white leaf. A beaker. Ethanol. Test tubes. Hot water. A source of bright light. Paper towels or filter paper. Procedures. 1. Take the healthy green leaf and the healthy white leaf and place them in separate test tubes containing enough ethanol to cover the leaves. 2. Put the test tubes in a beaker of hot water for a few minutes until the ethanol starts to boil. 3. Remove the test tubes carefully from the hot water and set them aside to cool. 4. Once the test tubes have cooled, use forceps or tweezers to remove the leaves from the ethanol and rinse them with water. 5. Place the green leaf and the white leaf on separate sheets of paper towel or filter paper and expose them to bright light for a few hours. 6. Observe and record any changes in color or appearance of the leaves. Results. The green leaf should have turned darker green as a result of photosynthesis, while the white leaf should have remained white. This demonstrates that chlorophyll, which is responsible for the green color of leaves, is essential for photosynthesis to occur. Without it, the white leaf cannot photosynthesize and therefore cannot produce glucose or oxygen. Practical activity. Aim, to investigate the necessity of light in photosynthesis. Give the requirements, procedures and results. Requirements. A healthy green plant with several leaves. A cardboard box or light-proof container. Black tape. Scissors. Water. A source of bright light. Procedures. 1. Choose a healthy green plant with several leaves. 2. Cover the plant with the cardboard box or light-proof container, securing it with black tape so that no light can get in. 3. Place the covered plant in a well-lit area. 4. Water the plant and wait for a few hours. 5. Remove the cardboard box or light-proof container from the covered plant and observe any changes in color or appearance. 6. Compare the uncovered and covered parts of the plant and note any differences in color or growth. Results. The part of the plant that was covered and received no light should have shown little or no growth and may have had yellowing or stunted leaves. The uncovered part of the plant should have shown signs of growth and leaf color change due to photosynthesis. This demonstrates that light is necessary for photosynthesis to take place and that without it, the plant cannot produce glucose or oxygen. Practical activity. Aim, to investigate the use of light in photosynthesis. Give requirements, procedures and results. Requirements. A healthy green plant with leaves. 2 beakers or glasses. Water. Sodium hydrogen carbonate baking soda. A source of bright light. A piece of black construction paper. Scissors. Procedures. 1. Fill 2 beakers or glasses with the same amount of water. 2. Dissolve a small amount of sodium hydrogen carbonate in one of the beakers or glasses to make a solution. 3. Cut a piece of black construction paper to fit over one of the beakers or glasses. 4. Place the healthy green plant in the beaker or glass with plain water. 5. Cover the other beaker or glass with sodium hydrogen carbonate solution with a black construction paper. 6. Place both beakers or glasses in a well-lit area. 7. Wait for several hours, ensuring that the plants are exposed to the same amount of light. 8. Observe and compare the plant's color and appearance, noting any differences. Results. The plant in the beaker or glass with sodium hydrogen carbonate solution and covered by the black construction paper should appear less green than the plant in plain water. This is because photosynthesis requires light, and the covered plant was not exposed to light, preventing photosynthesis. The plant in plain water should appear greener, indicating that photosynthesis took place since the plant was exposed to light. This demonstrates that light is necessary for photosynthesis to occur. Practical activity. Aim, to investigate the use of carbon dioxide in photosynthesis. Give the requirements, procedures and results. Requirements. A healthy green plant with leaves. 2 soda bottles or jars. Water. Sodium hydrogen carbonate baking soda. A source of bright light. A piece of plastic wrap. A straw. Procedures. 1. Fill 2 soda bottles or jars with water. 2. Dissolve a small amount of sodium hydrogen carbonate in one of the soda bottles or jars to make a solution. 3. Place the healthy green plant in the soda bottle or jar with plain water. 4. Cover the other soda bottle or jar with sodium hydrogen carbonate solution with a piece of plastic wrap. 5. Use a straw to blow into the soda bottle or jar with the plastic wrap, filling it with carbon dioxide. 6. Place both soda bottles or jars in a well-lit area. 7. Wait for several hours, ensuring that the plants are exposed to the same amount of light. 8. Observe and compare the plant's color and appearance, noting any differences. Results. The plant in the soda bottle or jar with plain water should appear less green than the plant in the soda bottle or jar with sodium hydrogen carbonate solution. This is because photosynthesis requires carbon dioxide and the plant in the soda bottle or jar with plain water did not have it. The plant in the soda bottle or jar with sodium hydrogen carbonate solution should appear greener because it had carbon dioxide, which is necessary for photosynthesis to occur. This demonstrates that carbon dioxide is necessary for photosynthesis to occur. Storage organs. 1. Storage in leaves. Leaves can sometimes act as storage organs for plants, especially in succulent plants such as aloe vera and Crassula ovata jade plant. In these plants, the leaves are able to store water, which helps the plant to survive periods of drought. Additionally, in some plants, such as onions and garlic, the leaves dry up and form a papery outer layer which serves as a protective covering for the bulb or corm, which is the actual storage organ of the plant. In summary, while leaves are not commonly used as storage organs in plants, they can play an important role in plant survival in certain species. 2. Storage in seeds and fruits. Seeds and fruits are important storage organs in plants. They are used to store nutrients essential for the germination and growth of the new plant. The nutrients stored in seeds and fruits are in the form of carbohydrates, proteins, and lipids. Seeds. Seeds are the reproductive structures of plants that contain an embryo, which develops into a new plant. They are often enclosed within a protective outer coating such as a hard shell or a fruit. Inside the seed, the embryo is surrounded by the endosperm, which provides nutrients for the developing seedling until it can photosynthesize on its own. Examples of seeds used for food and oil include legumes, grains, nuts, and oilseeds. Fruits. Fruits are the mature ovary of a flower and can take many different forms. They can be fleshy, such as tomatoes or apples, or dry, such as nuts and grains. The fleshy fruits contain seeds that are surrounded by a nutrient-rich pulp, while dry fruits often have seeds enclosed within a hard protective covering. Fruits provide a mechanism for seed dispersal and nutrition for animals and humans. Examples of fruits used for food include apples, oranges, bananas, and berries, while examples of dry fruits include peanuts, almonds, and sunflower seeds. Overall, seeds and fruits are essential for plant reproduction, as well as providing important nutrients to animals and humans. 3. Storage in stems. Stems can also act as storage organs for plants, storing nutrients in specialized tissues. In some plants, such as asparagus, the stem acts as a storage organ for carbohydrates and other nutrients during the dormant winter months, which are then used to support new growth in the spring. In cacti and other succulent plants, the stem is adapted to store water and nutrients to help the plant survive periods of drought. Additionally, some plants produce specialized storage structures called tubers, which are thickened, fleshy underground stems that can store significant amounts of carbohydrate reserves. Examples of plants with tubers include potatoes, yams, and sweet potatoes. In summary, stems can play an important role as storage organs in plants, with some storing nutrients or water to help the plant survive during times of stress or dormancy, while others produce specialized structures such as tubers for long-term storage. 4. Storage and roots. Roots are a common storage organ for plants and serve as an important mechanism for survival during the plant's life cycle. The type of storage and roots depends on the plant species and the environmental conditions. In some plants, such as carrots, beets, and radishes, the roots are enlarged and swollen to form a specialized storage structure called a taproot. These taproots store carbohydrates, proteins, and other nutrients, which are used to support the plant during its life cycle. In other plants, such as sweet potatoes and cassava, the storage is in the form of modified adventitious roots, known as storage roots. These roots accumulate large amounts of carbohydrates and can grow quite large, providing a source of food when other food sources may be scarce. Finally, in some desert plants, such as cacti, the roots can store water and nutrients to help the plant survive during extended periods of drought. Overall, roots play an important role as storage organs in plants, providing nutrients and water to support growth, survival, and reproduction. Advantages of storage organs. There are several advantages to having storage organs in plants, including 1. Survival during adverse conditions. Storage organs allow plants to survive during adverse conditions, such as drought or low light periods, by providing a source of nutrients that can be used to support their growth and reproduction. 2. Reserves for growth. Storage organs provide a reserve of energy and nutrients that can be used by the plant to support growth during periods of rapid growth, such as when new leaves or flowers are developing. 3. Seed production. Storage organs, such as the endosperm in seeds, provide nutrients to developing embryos to ensure successful germination and establishment of the new plant. 4. Human and animal consumption. Many plants with storage organs, such as fruits, vegetables, and nuts, are important sources of food for humans and animals. 5. Stabilization of plant productivity. Storage organs help stabilize plant productivity by buffering against fluctuations in nutrient availability and ensuring a steady supply of resources for growth and reproduction. Overall, storage organs are important adaptations that allow plants to survive in a wide range of environmental conditions and provide a source of sustenance for humans and animals. Revision exercise. 1. What is photosynthesis? Photosynthesis is the process by which green plants and some other organisms convert light energy from the sun into chemical energy in the form of glucose or other organic compounds. This process involves the absorption of light by pigments called chlorophyll, which is found in the chloroplasts of plant cells, and the use of that energy to drive a series of chemical reactions that ultimately result in the formation of glucose and the release of oxygen into the atmosphere. 2. List 2 characteristics of the palisade layer. Here are 2 characteristics of the palisade layer. 1. Cell shape. The cells in the palisade layer are elongated and columnar in shape. This shape allows them to pack tightly together and maximize the amount of surface area available to capture sunlight. 2. Chloroplast content. The palisade layer is rich in chloroplasts, which are specialized organelles where photosynthesis takes place. The high density of chloroplasts in the palisade layer allows for efficient capture of light energy, which is converted into chemical energy through photosynthesis. 3. Where do the following stages of photosynthesis take place? A. Light stage. B. Dark stage. The process of photosynthesis can be broadly divided into 2 main stages. A. Light-dependent stage. The light-dependent stage of photosynthesis occurs in the thylakoid membranes of the chloroplasts. This stage involves the absorption of light energy by pigments such as chlorophyll, which creates high-energy electrons that are used to generate ATP and NADPH. Oxygen is also produced during the light-dependent stage as a byproduct of water being split. B. Light-independent stage. The light-independent stage, also known as the Calvin cycle or dark reactions, occurs in the stroma of the chloroplasts. This stage involves the use of the products of the light-dependent stage ATP and NADPH to convert carbon dioxide into glucose through a series of enzyme-catalyzed reactions. Unlike the light-dependent stage, the light-independent stage does not require direct absorption of light energy, but it does depend on the products generated by the light-dependent stage. 4. Briefly explain how temperature affects the rate of photosynthesis. Temperature can affect the rate of photosynthesis as it directly influences the activity of enzymes involved in the process. Generally, a moderate increase in temperature can increase the rate of photosynthesis up to a certain point, after which it begins to decrease. At low temperatures, enzymes involved in photosynthesis have low activity and the rate of photosynthesis is slow. However, as the temperature increases, enzyme activity also increases, leading to an increase in the rate of photosynthesis. But if temperature becomes too high, above the optimal temperature range for the plant's enzymes, the enzymes lose their shape and functionality, and denaturation occurs, which can lead to a decrease in photosynthesis rate. Different plants have different optimal temperature ranges for photosynthesis due to differences in the activity of their enzymes. For example, some plants have evolved to perform best in cooler temperatures, while others are better adapted to warmer temperatures. Additionally, temperature can indirectly affect photosynthesis by influencing factors such as the availability of water, which is also necessary for photosynthesis to occur. 5. Give any 2 advantages of storage organs in plants. Certainly. Here are 2 advantages of storage organs in plants. 1. Provides a source of energy. Storage organs in plants such as potatoes, beets, and carrots store large amounts of carbohydrates such as starch and sugars. These carbohydrates are used as a source of energy for plants during times when photosynthesis cannot occur, such as during droughts, winter, or when the plant's leaves are not actively photosynthesizing. This energy can also be used to support growth and reproduction. 2. Enables survival in unfavorable conditions. Storage organs offer a way for plants to survive unfavorable environmental conditions. During periods of drought, for example, plants can rely on stored water in their storage organs to survive until they can obtain water from the environment. Additionally, some plants store defensive compounds in their storage organs that can protect them from predators or pathogens, providing them with an evolutionary advantage in their environment.